So hello everyone, and yeah, this is our first now video, and this is our topic, cancer. Yeah, cancer, pretty great topic. I guess for first video, and it's pretty experimental thing we are trying. And cancer is basically, I guess, yeah, it's a testing thing, but we will see what cancer is. So cancer is a microevolution process. Oncogenes, yeah, we'll see which genes can act, like which genes can form cancer or malfunction of which proteins, enzymes, pathways can cause cancer. So let's start here. So this is a first slide. Ignore this. I have downloaded it from somewhere. But so yeah, cancer is a microevolution process. So you can read here. It is given. Uh, cancer cells break most of basic rules of behavior by which multicellular organisms are built and maintained because they are undifferentiated, proliferative, and uh, they have that dominance over other cells, and they basically weaken every single system. They affect like tumors. Most of there is some terminology, a tumor, that is abnormally growing cells, and tumor is benign. Yeah, when it is mostly considered in a clinical way, grade 1 and grade 2 cancers, tumors are benign. And after grade 3, 4, those are malignant, which invades the tissues, spread through our body, through the blood. Blood cancer is called leukemia. It's one of the most common cancer, blood, prostate, breast cancer. And yeah, carcinoma and this all, it's a physical cancer. It's easy and yeah, the most important thing, principle of oncogenesis. So what is this? Principle of oncogenesis. So cancer, of course, arises from a single cell initiated by somatic mutation, as you can see here. And these they have gave three different important reasons which can cause mutations in DNA and carcinogens. Yeah. So first is chemical. Yeah, natural synthetic chemicals. Few I can tell as most of ROS generating chemicals. They can cause cancer. H2O2 being one of them. Second is physical. UV radiations. Uh, of course, radiation can cause mutations, as you know. It's both like UV radiations are used to cause mutations in plants for better well-being of plant, to activate some genes, and but in mammals, animals can cause cancers. Biologically, there are tumor viruses, pathway failure, system failures. Tumor viruses here I would like to emphasize one of the most important virus. Currently, it is called. Please ignore my handwriting. It's first time I'm writing. It's HPV 16, and yeah, this is the root cause of I would say cervical cancer. Like this virus 100% causes cervical cancer if it affects anyone, mostly females. Yeah, and it's so crucial to basically vaccinate young girls because the vaccine which is currently being developed in India by Serum Institute, as per them, if the vaccine is given in the age of 8 to 16, that is the most effective thing. So this is the most important. This is the root cause for death of millions of people, females, and these are some mutations, point deletion, application. Yeah, that's easy. And this is the basic principle for that. As we can see, deletion of one mutation, single point like single base pair changed, deleted, right, while many processes. Gene amplification is the chromosome rearrangement, overproduction of proteins. You can see here. This all thing it can basically over oncogene, like it starts the oncogene, you can say. And two ways of amplifying oncogene: dual chromosome containing mycelium gene, dual copies of MYC on the same chromosome. This is experimental data. I don't think it's that much needed. Yeah, it's okay. We'll see this. Okay, this is important. So, what's P53? So, what's P53? P53 is a signaling protein. So basically, you all have studied biology at least class 10th or 12th, so you must know that there is this cell division which happens continuously in the body. There's a continuous cell division, and in a cell division where we see that uh, cells divide, and but there is one thing: cells divide. There are regulatory pathways. Like if there is this path of cell division, there is a regulatory pathway which regulates the cell division. And what we saw that cancer is a proliferative cell, undifferentiated proliferative cells, or continuously dividing cells, which are not regulated. So there is problems in regulation, and these are caused by p53. This is a tumor suppressor gene, and if this protein is not present, regulation will not happen. So basically, there is regulation. Basically, there's a simple mechanism that is, firstly, cycle goes into, like, there are four phases we know: G1, S, G2, and M phase. When there is a transformation from G1 to S phase, there is a gate. It is known as cyclin CDK. And when this complex is phosphorylated, then only a cell can enter the another phase, a rest phase. And the requirement is synthesis of all chemicals required for next phase. But what we see is that if it's not completed, if there are the nutrients are not needed, then cell goes back. It goes back for cell senescence. It gets one more chance to do it. See how great our world is. It gives chance for everyone. Why can't we give chance for us? Like, yeah, sorry. And this chance, and after the cell senescence, the again, if the nutrients are not fulfilled, then our cell signals p53. This proteins signals apoptic proteins. This is the main link between them. This is the protein which acts as a tumor suppressor. This signals apoptic protein, and apoptic proteins further act uh, against these cells to kill the cells. And again, if apoptic proteins are increased, they can even kill the normal cells. But for that, we have anti-apoptic proteins to keep a control of apoptic proteins. And if there are anti-apoptic proteins are high, concentration of anti-apoptic proteins is high, then apoptic proteins won't kill the tumor cell. So you see how crucial it is, how perfectly it's regulated and tuned. So in a mutation can inactivate a tumor suppressor gene. So if it is inactivated and we see something of this happens, it won't get killed, and excessive proliferation will start, and this will develop tumor. See, only 1 in 30,000 normal people develop tumor, but it's changed because of the current lifestyle mostly. And this is genetics, and all this is a simple process, multi-step process. So accepted position of mutant cell. This is a mutant cell formation, cell mutation start, and dangerous cell proliferation. So this is the catalyst of penicillin tumor, and when it grows and when it like grade 1, grade 2, and when it enters the malignant stage, it flows through our blood and many other pathways to the various tissues, and yeah, metastasis happens. And yeah, it's very, and there are a lot of therapies I would actually like to tell, like cancer therapies currently new, which are immunotherapy by Dr. Patrick Soon-Shiong, and a newly developed RT therapy. And yeah, these are pretty great therapies currently for cancer. And I guess I should stop here because this is our first video and it's almost 9 minutes of me lecturing on some boring cancer, going into details. But I guess, and first of all, thank you if you have stayed till here. And yes, I know there have been a lot of mistakes in this video, but I I'm really sorry for the mistakes. But we are just starting, and we will improve. Will improve. Yeah, thank you.